DNA replication is the process by which a cell makes an identical copy of its DNA, ensuring that genetic information is passed on to daughter cells during cell division. It begins at specific locations on the DNA molecule called origins of replication. The enzyme helicase unwinds the double helix, creating two single-stranded templates. Each template strand is then read by DNA polymerase, which adds complementary nucleotides to form a new strand. The leading strand is synthesized continuously in the direction of the replication fork, while the lagging strand is synthesized in short segments called Okazaki fragments, which are later joined by the enzyme DNA ligase. This process occurs in the 5-3 direction. Additionally, RNA primers are laid down by primase to provide a starting point for DNA polymerase. The result is two identical DNA molecules, each composed of one old strand and one newly synthesized strand, ensuring accurate transmission of genetic information.